Today we are talking about eukaryotic cells. These are eukaryotic cells. Does anybody know what kind they are? For those of you who've already had lab this week, you looked at these under the microscope. They didn't look as cool because we had a different species. Not desmids, diatoms. These are diatoms. And it was a Victorian art to go out and collect these organisms which live in aquatic habitats, saltwater and freshwater, and go under a microscope and look at how cool these different organisms are, their different shapes, and arrange them into beautiful patterns. The way that you move these guys around under a microscope is you typically get a hair from a pig, so a hog hair, and you use that to move these microscopic organisms around. These organisms and some of the other ones we are going to talk about today actually form a hard shell, so a glass silica shell that remains even after the cell is dead, and that's why you can make this cool artwork. So these are all old diatoms, but they have very, very diverse cell structures. So today we're talking about eukaryotic cells. And eukaryotic cells are cells that make up humans. They're cells that make up plants. There's a lot of single cell or unicellular eukaryotes as well. And one of the things that makes eukaryotic cells different from archaea and bacteria, which are the prokaryotes, which we talked about on Tuesday, is the fact that they can fold their membranes, which have then turned into membrane-bound organelles. So if we're looking at these pictures, here we have, this is actually the lining of a mosquito stomach. So these pictures are pictures I took when I was in undergrad as an undergraduate research student. So here are cells. You can see the nucleus of the cell. This is actually blood that's inside the mosquito stomach. And as we zoom in, you can see all of this folding that's happening. You can see these membranes. And that's what differentiates, one of the things that differentiates eukaryotic cells from the prokaryotic cells. So today is focused on domain eukarya. And they have internal compartmentalization. They have DNA inside a nucleus, membrane-bound organelle. The nucleus and the endomembrane system most likely formed from membrane folding. So we had membranes of our cells that folded in and separated and then became different organelles. First clicker question. So a different way to look at this exact same tree, we can see here's our eukaryotes. They are more closely related, because this is where they join, to the archaea than they are to bacteria. So eukaryotes do energy metabolism in the mitochondria and the chloroplast. So think back to last semester, you learned about cellular respiration and photosynthesis. That is where eukaryotic cells do their energy metabolism. Mitochondria and chloroplasts have their own DNA. Which means that they originally were their own free-living organisms. So it is hypothesized that mitochondria and chloroplasts gained entry into early cells by endosymbiosis, which means that there is a symbiotic relationship that formed between the cells that were originally mitochondria and chloroplasts and the early eukaryotic cells. So you can see here, one type of bacteria gave rise to chloroplasts, and a different type of bacteria gave rise to mitochondria. Now, if we think about eukaryotes, we're very diverse. So this endosymbiosis that happened very, very, very early on. Oops, no, go back. And there's two hypotheses as to how this happened. So the first hypothesis is that you had some ancestral archaeon-like cell, and here's the DNA in the cytoplasm, and the infolding of the plasma membrane happened first, which helped to form the nucleus, and then later a proteobacterium was engulfed, and that is what became, in this example, the mitochondria. So that's one hypothesis. Infolding and formation of the nucleus happened first, and then we engulfed the endosymbiotic ancestral mitochondria. The other hypothesis is that there was an endosymbiotic relationship before infolding of the membrane happened. So the proteobacteria was an endosymbiont in an ancestral archaeon cell, and then later infolding happened that formed the nucleus. So we still are not sure in which order this happened. But based on looking at DNA, we know that our mitochondria have bacterial ancestry. Any questions on this? So many organisms in the domain Eukarya are multicellular. And there are advantages to being multicellular. And this EG means for example. So for example, being multicellular helps you to avoid predation. Animals, for example, can run away from predators. If you're multicellular and you're a plant and an organism bites part of you, the rest of your body is okay. So it helps you to avoid predation, it helps you to survive longer. Also, it helps you to maintain a position on the surface, so like a terrestrial, ground, land environment, or in water. So for those of you who have had lab already today or yesterday, you looked at a lot of different eukaryotes and some prokaryotes. And what did you notice about some of those organisms you looked at when they were moving? Did they move fast or slow? Oh, it depended, but a lot of them like zipped around, right? Think about the rotifer. How fast did the rotifer move? It moved really fast. If you're smaller and you don't have many cells in your makeup, you're going to be moving a lot faster. You're going to be pushed around by, if you're in the water, by currents. If you're in the terrestrial environment, you could be blown around by the air. You could be picked up. Anytime you touch something, you are picking up unicellular organisms on your hand. So if you're multicellular, it's harder for you to be moved places you don't want to be moved. So those are just some examples of how there's advantages to being multicellular. Most of the organisms we are going to focus on today are either unicellular or they have what is called simple multicellularity. We have advanced multicellularity as humans because our cells have specialized function and they work together as a whole. If you took our heart out of our body, it would die. That cannot function on its own. It needs that intricate system that has evolved over time. But many of these organisms can be a single cell and survive. So in simple multicellularity, adjacent cells stick together. They just stick to each other. They don't have a lot of communication with each other. They're more sticking together for protection, or there's a good nutrient source in the area. So they're not actually transferring many nutrients to and from each other. They're just all there to get their own nutrients. Almost all of those cells in that group are not differentiated. They just have the same function. All the cells in that group do the same thing. They have to carry out all the different functions to survive. And every single cell is connected with the external environment that they're living in. We have a lot of cells in our body that never, ever, ever, hopefully ever touch the air. It's really bad if they do. But in simple multicellularity, all of the cells are touching the external environment. And these are pictures of examples of simple multicellularity. 36 of the 119 eukaryotic kingdoms exhibit some form of simple multicellularity. So this is a common way to survive. So today, the groups that we're really going to focus in on are the protists. So we'll spend more time talking about plants. We'll spend time talking about fungi. That will be our first lecture after our exam next week. And then we'll spend a lot of time talking about animals. So today we're really focusing more in on these protists that we don't spend as much time talking about because a lot of them are more simple organisms. So we can see here on our tree, we have bacteria. Here's where our chloroplasts and mitochondria came from. This is a historical idea of how organisms are related to each other on this particular tree. Guess what time it's for? So we're going to spend the rest of class talking about eukaryotic diversity, and I will move that clicker thing. So in eukaryotes, there are 7 what we call super kingdoms. Okay, so there's 7 super kingdoms that we use to classify organisms that are similar to each other. Every super kingdom is a monophyletic group because it represents a clade. So if we're looking up here at the opisthecons, animals, choanoflagellate, and fungi all form one monophyletic group. They all share a common ancestor. So every super kingdom is a monophyletic group. Any group that's in green is a group that has photosynthesis. Not every organism in that particular group may undergo photosynthesis, but there are at least some, which tells us that photosynthesis evolved multiple times in eukaryotic cells. So that endosymbiotic relationship with chloroplasts happened multiple times through the evolution of eukaryotic groups. So I'm going to go through each of them separately. Yeah.
Why is the one branch dotted?
That's a great question. Why is this one branch dotted? Does anybody know? So we're not quite sure, and these are newer organisms, some of them we didn't really know about. We haven't done molecular testing to figure out where they belong. Okay. So our first group are the opisthecons, or opistheconta. Opisthecon is Greek for posterior pole. So the major defining characteristic of opisthecons is that they have a single posterior flagellum. So if we look at choanoflagellates, they all have a single posterior flagellum that helps them move and swim through their environment. Animals, a bunch of smaller organisms, and fungi are all in the superkingdom Opisthecanta. So we are opisthecons. So you could go home and tell your friends and family, did you know you're an opisthecont? And hopefully you can giggle and be like, I know something you don't know. Choanoflagellates are our closest relatives. So choanoflagellates, these little single-celled organisms, are our closest relatives to humans. They tend to have a single flagellum, and they have a little ring of microvilli, and they actually are closely related to sponges, which are the most basal or basic ancestor of all animals. And they're similar to sponges. Opisthecons are the most diverse superkingdom in all of the eukaryotes. I mean, just think about how many different animals there are. Think about how many, you probably don't even realize how many different fungi there are. You will next Thursday. 75% of all described organisms on the planet Earth fall within this superkingdom. So I said animals are opisthecons. So that means humans are opisthecons. Does anybody know what cell we have, that humans have, that has this defining characteristic, that propulsion is done by a single posterior flagellum? Yeah. Sperm. So sperm is the only cell that most mammals still have, or most animals have, that still has this particular defining characteristic. This is a picture of Microsporidia, and it is a closely related organism, closely related to animals, because they're in the same superkingdom, and they actually cause internal parasites and diseases. And this is a picture of it bursting out of a intestinal cell. So our next group are the amoebozoas. So if you've had lab already, you hopefully got to see an amoeba. If you haven't had lab, you will get to see an amoeba. So amoebozoas obviously include amoebas and other organisms. Amoeba's defining characteristic is that they have what is called a pseudopodia, and that's used for locomotion and feeding. So on a typical amoeba, the pseudopodia are these finger-like projections, and they use those to reach out, and they can grab food and have food diffuse into their cells, or it helps them move through the environment. They are usually unicellular, but there are things called slime molds that can be multicellular. So in plasmodial slime molds, which is this pretzel slime mold and this slime mold here, they become multicellular before producing spores, which are their reproductive gametes. So they produce these little stalks in this particular slime mold, become multicellular, and then they release themselves into the environment. In cellular slime molds, so this clear-looking organism, they form multicellular aggregations or groups when there's low nutrient levels. So they'll form together, so it makes them easier to move throughout the environment to find food. Some cellular slime molds also will produce little clusters of multicellularity that produce spores. And some of these amoebozoas actually cause dysentery, which is an infection of your intestines, causes you to have severe diarrhea. There's usually blood and mucus in your feces, and if you don't get treated for it and you don't get enough fluids, you die of dehydration. Closely related to us, though. The next group are the archaeplastids. These are photosynthetic organisms, so everybody in this group goes through photosynthesis. They all have chloroplasts. And we believe that everybody in this group, which includes red algae, green algae, land plants, they acquired their chloroplasts through primary endosymbiosis, and evolutionary biologists believe that they are the direct descendants of the protists that first evolved this. So they were the first group of organisms in eukaryotes to have endosymbiosis with chloroplast. They are the second most diverse group after the opisthecons for all eukaryotes. Land plants, which are in these groups, which we'll spend a few lectures talking about in a few weeks, land plants dominate the eukaryotic biomass on the planet. So by biomass for eukaryotes, land plants have the most. If you think about that, think like a redwood, a giant tree, there's lots of plants everywhere. They have the most biomass of all eukaryotes. Not of all organisms. Bacteria and archaea would outweigh them by a lot. There's over 400,000 species of described land plants. Stromenopelia is our next super kingdom. These guys, this is a simple version. This is a simple Stromenopelia. They have a motile stage flagella, and it's covered in hollow hair-like structures. So they have a hairy flagellum, and a lot of them have a second smooth flagellum that tends to be shorter. So their flagellum has little hairs that are coming off of it. There are 3 major subgroups. There are water molds, which caused the Irish potato famine. So it causes potato blight and other blights on plants, usually vegetables and starchy foods that we eat, makes it so we cannot eat them. So water molds were responsible for the Irish potato famine. We have diatoms, which here's a few diatoms down here. They have a silica, almost like glass, internal structure. I showed you a bunch of pictures of them in the beginning of class because they're so cool. And brown algae. So brown algae is not a plant, it is a Stromenopelia. An example of a brown algae are giant kelp. And there's a picture of a seal just for you to get an idea of how large giant kelp is. It is giant. So most of the organisms in Stromenopelia are unicellular. They're very small. You have to look under a microscope to see them. Brown algae, that is not always the case. In the case of giant kelp, they actually have developed distinct organs for attachment. So they have distinct parts of their body that help them to attach to the ocean floor. They have distinct organisms, so leaf-like structures that help them photosynthesize. They have made these flotation devices. They've evolved over time to help them float, and they have evolved reproductive organs. So in this particular example of brown algae, we actually have cellular differentiation. And otters, in case you were curious as to why there's an otter there, are very important for the health of giant kelp. And otters are cute. So giant kelp is an extreme example of Stromenopelia. Next, we have Alveolata, and their defining characteristic is that they have a cortical alveoli, or sacs, beneath their plasma membrane that function just as our Golgi bodies do. So that's their defining characteristic. There are 3 major subgroups: Dianaflagellates, which are typically photosynthetic, and they have 2 flagella. So flagellum is singular, flagella is plural. So you could see here, it's pointing to their 2 flagella that's coming off. We have ciliates, which are interesting because they have 2 nuclei per cell. And then we have the apicomplexans. And the reason we care about the apicomplexans is that is the group that malaria parasites are in. So the parasites that cause malaria are eukaryotes, which makes them very different than most other diseases that mosquitoes can spread. So malaria is in the superkingdom Alveolata. Next we have Rhizaria, and their defining characteristic is they have a very slender pseudopodia used for locomotion. A lot of these have a hard outer shell, usually made of silica, just like diatoms. And you can see here's their slender pseudopodia sticking out. You can see them sticking out in these fossilized groups, and it helps them with locomotion. So in amoebozoa, it's a big fat pseudopodia. In Rhizaria, it's a very slender, delicate pseudopodia. And last but not least is excavata. So excavatas are kind of weird. Many excavatas have unusual flagella. So you can see here this organism, Giardia, which causes Giardia, has many flagella sticking off of it, and they have altered mitochondria that cannot generate ATP. So they have to provide ATP to their mitochondria. Many also have this groove along the side of their shell or cell. You could see this groove here. So those are defining characteristics. Yep.
It doesn't make ATP. What does the mitochondria do?
We're not sure. What we think happened is that it doesn't use the mitochondria, but that is still in there just because if you look at the relationship, it's further out, it's more derived, it's a newer group compared to some of these other groups, and so it's probably just there kind of as a remnant. Yeah.
What does Giardia affect?
What does Giardia affect? Does anybody know what Giardia affects? It's also just a really nasty intestinal disease.
So it doesn't go away once you have it?
It does. You could treat it. So we just went through the 7 super kingdoms of eukaryotes.